My name is Randy McIntosh. I'm the director of the Institute for Neuroscience and Technology, also professor in BPK, and amateur musician and poor runner, but otherwise pretty good. I'm very happy today to have Jennfer Ferris here with us. She's currently doing a postdoc in gerontology and with the BC Observatory, for Population Public Health, BC Centre for Disease Control. She has a degree in rehab from UBC, and now she's focusing on expanding her domain to look at cardiovascular risks and brain health together. And we'll hear about that now.
Thank you. Thank you so much. Thank you so much, Randy, for the invitation, and Randy and Kelly for organizing everything. And thanks to all of you. I'm really excited to be here with you today. The goal for today is just to present an overview of some of the work that I've been doing and also just some ideas that I've been thinking about. And I'm hoping to hear from all of you at the end of this as well, and some of your ideas and input on these concepts. So I'll just start by acknowledging that I work and live on the traditional lands of the Coast Salish people, and in particular, the work today has been conducted on the traditional, ancestral, and unceded land of the Musqueam, Squamish, and Tsleil-Waututh nations. So starting with a general intro, I'm sure this will be familiar to many of you in the room, but we're going to just start by discussing cerebrovascular disease broadly. And so generally, when you say the term cerebrovascular disease, what comes to mind is stroke. And stroke, I'm going to define here as large-ish vessel disease. So the reason I have the ish there is because large vessel occlusion is a specific type of stroke. So I'm not saying that this is all large vessel occlusion stroke, but instead a stroke is a vessel disease that's impacted a vessel that's large enough in order to cause a disruption in function. There's two major subtypes to stroke. So stroke can be caused by either a clot, which is an ischemic stroke, or a bleed, which is a hemorrhagic form of stroke. And ischemic strokes make up the vast majority of strokes. There's about 90% of stroke events are ischemic. So just some general numbers and statistics around stroke in Canada. This is a little infographic from the Heart and Stroke Foundation. So currently there's around 400,000 Canadians living with the effects of stroke. And from some of the data that I'm working with in the BC Chronic Disease Registry through the BC Centre for Disease Control, this translates to about 3% of older adults in BC who are currently living with the effects of stroke. And this is a really significant condition because around half of the people who survive a stroke will need help with their daily activities. So they'll have some form of chronic impairment as a direct result of the stroke. With advances, oh, I'm sorry. Let me go back. There we go. There's been a lot of significant advances in acute stroke therapies and treatments, which means that more people survive strokes and stroke outcomes have generally improved. But even in this context, we have a lot of lasting impairment to deal with. So rehabilitation is extremely important in the context of stroke. Okay, so I introduced stroke as a large-ish vessel disease, and maybe you're wondering, what about small vessels? If not, I'm going to pose that question. And so certainly there's many other subtypes of cerebrovascular disease that impact more of the small vasculature of the brain. And we can consider an umbrella term for these different pathologies called cerebral small vessel disease. So showing here some, just a range of the subtypes of cerebral small vessel disease. These are guidelines from the recent STRIVE-2 updated criteria for classification of cerebral small vessel disease. There's a lot to look at here, but essentially the idea is to give you an overview of the many different forms of small vessel pathology that are detectable. And a few things to note. So number one, these are really defined by neuroimaging features. So at the bottom, you can see on different types of MRI sequences, whether signal is increased or decreased or isointense. Looking at these sorts of imaging features is what neurologists and clinicians can use to distinguish between these different forms of small vessel disease. So that's one thing to note. And the other thing to note is that these are also defined by size. So there is no clear really boundary between small vessel disease and overt stroke. And in fact, some of the subtypes of small vessel disease, let me just grab my mouse here. So some of them are essentially ischemic infarcts. So you might have a small subcortical infarct or a lacune, which can sometimes be an ischemic stroke and sometimes is a area that's cavitated within otherwise unhealthy brain tissue. But what's defining them is essentially their size. So, a small subcortical infarct would be defined as an infarct that's less than 20 millimeters. So again, there's quite a range of different forms of small vessel disease, but for the rest of the talk today, I'm really going to focus in on white matter hyperintensities. And white matter hyperintensities, all it means is an area in the white matter that is hyperintense or bright on a T2 weighted image. So FLAIR imaging being a really common form to assess this. And white matter hyperintensities are significant in this context because they're one of the most common forms of cerebral small vessel disease and that can be easily detected on MRI. And the other thing to note is that these are often one of the most common incidental findings on MRI. So giving an image here of some different types of white matter hyperintensities and some of the typical progressions that we see with white matter hyperintensities, they tend to start around the rims of the lateral cerebral ventricles in a periventricular distribution. And then they gradually extend outward into the deep cerebral white matter and ultimately become confluent or sort of merging between deep and periventricular lesions. They're also fairly symmetrical in their distribution, but not always. And what we see is in the course of this disease progression, the white matter hyperintensities are strongly associated with cognitive and functional decline. And they can also be categorized under a larger umbrella of vascular cognitive impairment, which includes small vessel disease as well as stroke-related cognitive impairment. And when I say they're quite common, you might ask how common, and we don't really know the answer to that because ultimately this is something that's detectable on neuroimaging. And so we don't have really good normative demographic distributions of these, but in large studies that have been done, we can say there's probably anywhere from 40 to 90% of adults who are aged 60 and older who show some evidence of white matter hyperintensity on T2 imaging. So extremely common and also strongly associated with functional decline in aging. Now, moving on to the broader context of cerebrovascular health, we know that stroke and cerebral small vessel disease, they have a lot of overlap. They're really part of a similar cerebrovascular continuum, and they share common risk factors. There's a number of different cardiovascular risk factors, but some of the large offenders are here, including age, high blood pressure, diabetes, heart disease, and high cholesterol. And these are also extremely common, and they have these sort of synergistic negative impacts for brain health and overall health. And so Heart Stroke Foundation suggests that about 90% of individuals in Canada have at least one risk factor. This doesn't only include these medical condition risk factors, but also behavioral and lifestyle risk factors. And this is increasing risk of future heart disease, stroke, and vascular cognitive impairment. The other thing is when we think about this idea of risk factor synergies, we know that having one of these risk factors, including these medical conditions listed here, can also increase your risk of developing another one. So stroke would be one of these conditions to consider. And in many cases, these might also be part of a similar etiological disease continuum. So we can think about cardiovascular and metabolic health very holistically as being something where you have these compounding either beneficial effects and opportunities for intervention and prevention, and also potentially negative synergistic effects for long-term health. So if we're thinking about white matter hyperintensities as a background state of your cerebrovascular brain health, the question that I've been asking in a lot of my research is how are white matter hyperintensities impacting trajectories after stroke? And a sort of a broader question is how they might impact the risk for and recovery from stroke. White matter hyperintensities themselves are an independent risk factor for stroke, controlling for these other cardiovascular conditions. And their role in stroke recovery is largely unknown to date. So I'm just going to move to sort of some concepts of resilience here and ask this question of what does it mean to age well? Well, what it might mean to age well is slowing decline over the course of aging. It might mean preventing a disruptive event like a stroke from occurring. Or if a disrupting event occurs, it might mean recovering rapidly. So I'm presenting here a framework which was developed in collaboration by Andrew Wister, my current postdoctoral supervisor, and some other colleagues, where they have this concept of resilience as being the ability to, in the event, can you see my, yes, okay. So we have this gradual decline with aging over time. And if there is some sort of critical event that occurs, some individuals who are showing good resilience might show a quick return back to a similar level of function as they had before the event, and other individuals might show a gradual decline. And so essentially resilience is this idea of trying to identify the factors that are characterizing these two subgroups and promoting good recovery or good resilience. Another conceptual framework of reserve and resilience that I've recently found useful was this paper that was published from Stern. And the reason that I liked this publication was because it really got into some theoretical detail around brain resilience specifically. So they define brain resilience as the capacity of the brain to maintain cognition and function in aging and disease, which is a pretty broad definition. But they get much more specific in this paper. They had 3 proposed mechanisms of brain resilience. This includes, first of all, brain reserve, which is defined as the neurobiological status of the brain at a single point in time. And the idea being that individuals who have higher brain reserve can tolerate more depletion to their system before symptoms emerge. And so putting up a little schematic here, if we think about a relationship between function and time, and there being some sort of threshold at which function declines such that symptoms are emerging, then brain reserve is sort of the distance from any individual to the symptomatic threshold at baseline. So the higher that you begin, the longer you have to fall before you show impaired function. There's also this concept of brain maintenance, which might be the absence of change in brain marker over time as a determinant of preserved function. So in other words, if brain reserve is thought of as the intercept between function and time, then brain maintenance might be the slopes, the speed at which you're declining over time. And finally, they propose this concept of cognitive reserve, which is what we often think about when we're thinking about resilience. And this is the idea that you can have performance that's better than expected, given the degree of injury or disease. This isn't a perfect chart of this concept, but essentially we might think of it as subgroups of individuals who have a capacity to moderate ongoing declining processes in the brain in order to preserve function. And I'm just going to elaborate because I'm going to spend time talking about concepts 1 and 3 in the rest of the talk. So, there's many frameworks, there's many different ways we can define these concepts, and this is one that's been used. I'm going to suggest that maybe we think about brain reserve as a brain buffer. So the idea that you have sort of money in the bank, so to speak, you have a well of brain reserve that you can draw on in the event of decline. And cognitive reserve, I'm just going to propose that we think of this as functional reserve. And the only reason for that is because I'm going to be talking a lot about motor systems and motor impairment. So this concept doesn't apply only to strictly cognitive domains. Okay, so where do white matter hyperintensities come in this framework? As I mentioned before, we can think of white matter hyperintensities as a visible manifestation or an end-stage manifestation of a progressive ongoing neurovascular process. And the thing that is interesting about white matter hyperintensities is they have this sort of insidious potential progression and onset. And so there's this idea that symptomology, the lesions themselves might be subtle or silent until you reach some sort of critical threshold, and then you have detectable change in behavior. And this means that there's a unique role in neuroimaging for the assessment and research of white matter hyperintensities. First of all, they're defined, as a neuroimaging feature, but also that you might have a lack of ability to objectively detect these in behavior when the disease process is very early. So how do we measure the severity of white matter hyperintensities? You often see visual rating scales applied. So the Physica scale is a common one where you just visually grade the severity of white matter lesions. And also there's work using volumetric analyses. So this was just a methods paper that I looked at where you try to automatically segment or manually segment white matter hyperintensities, and then you can measure their spatial distribution and their volume in the brain. Oh, there we go. So what are white matter hyperintensities is the question of the first bit of data I'm going to show. And what we know is that they're quite variable in their histopathology. And there's sort of a number of different theories, and this is an ongoing debate about how they actually form neuropathologically or what they mean in terms of the actual physical function of the brain. But the general idea is that you probably have some sort of disruption to blood-brain barrier, you have disruption to the vascular endothelium, and this is leading potentially to interstitial edema and fluid accumulation. And then over time, you might have astrocytes being affected, changing trophic support for neurons and glia, ultimately potentially damage to your myelin-producing cells, your oligodendrocytes, and then neuronal degeneration atrophy over time. The order of events isn't totally clear yet, and I'll also mention that there's a lot of focus on arteries in this research, but work from one of my mentors, Sandra Black, also implicates venous vessels as being important in white matter hyperintensity pathology. Essentially, you have this idea that there's an evolving pathology that's occurring. And so the question is, can quantitative neuroimaging capture these brain changes in white matter hyperintensity? So sorry, my animations are off here, but just giving a little introduction to diffuse and tensor imaging. This is a MRI-based technique that measures quantitative structures of brain microstructure. And it does so by applying models that measure directionality of movement of protons in a voxel, which is a 3D MRI pixel. So if you imagine a densely packed white matter bundle in this voxel in the brain, and your water proton, essentially your movement in this voxel is going to be restricted along two axes, but relatively unrestricted along the length of the axon. So fractional anisotropy, or FA at the top there, indexes the degree of directionality that you have in diffusion, and mean diffusivity is just an overall measure of how unrestricted the water proton is in all three dimensions. So if you're a dense white matter, healthy white matter bundle, then you will have a lot of directionality in the movement of water protons, and this indicates a way that we can infer white matter structure from diffusion tensor imaging. And the idea here being, in the context of stroke, as an example, if you're a neuron that experiences injury along its length, you're going to have ultimately a loss of structure in the neuron, fluid accumulation in the space that's damaged, and then this is going to decrease FA and increase MD. So this is the general principle behind DTI, to use to measure structural changes in the brain. So there's research using DTI in the context of white matter hyperintensities, and something interesting that's emerged is this idea of a white matter hyperintensity penumbra. So penumbra being a concept from the stroke literature, this is tissue that is under threat, but has not yet progressed to full death after an interruption in blood supply. And there was work that was done using DTI to measure spatial characteristics of white matter around the white matter hyperintensity lesion. And what you see, so this first image is just a schematic of findings from this 2011 study in stroke, where if you look at the core injury or the white matter hyperintensity lesion itself, they're using the term white matter integrity here to refer to fractional anisotropy. So you see FA gradually decline into the white matter hyperintensity lesion itself, and then gradually increase with increased spatial distance from the white matter hyperintensity. But the concept here is that in this penumbral or bordering zone to the lesion, fractional anisotropy is still measurably altered. And so the same thing was noted here in this more recent paper from Joanna Wardlaw's group, where if you look at FA in the white matter hyperintensity itself, it's low, and then it gradually increases with increasing distance from the white matter hyperintensity lesion. And MD shows the same pattern, but in the opposite direction. So again, MD being highest within the white matter hyperintensity and then gradually decreasing with increasing distance. And something interesting in this study was they actually looked at white matter tracts that weren't directly lesioned by the white matter hyperintensity. And you still saw the spatial gradient effects neighboring the white matter hyperintensity lesion. Another thing that's been done with diffusion tensor imaging is looking at tissue that appeared to be normal, healthy tissue at baseline, and then over the course of a follow-up time point, converted into a white matter hyperintensity. So this is work from Amy Brotman's group, where it's still based in diffusion tensor imaging, but they deconvolute the signal of the diffusion tensor into a white matter component, which is the W, a gray matter component, and a CSF-like component, which is in red. And then along the x-axis here, what you see is this would be normal appearing white matter that remained normal appearing white matter at a 1-year follow-up. Then you have white matter hyperintensities at baseline that stayed white matter hyperintensities at a follow-up. And then the interesting part here is normal appearing white matter that then went on to convert into a white matter hyperintensity at follow-up. So what you see is in your normal appearing white matter, the white matter-like signal and gray matter-like signal is clearly different from your CSF-like signal. In the white matter hyperintensity, CSF-like signal is higher, and the white matter, gray matter, and CSF-like signal are not as easily differentiated. And in tissue that was normal appearing at baseline, but then went on to convert, it actually looks very similar to those persistent white matter hyperintensities. So perhaps indicating some sort of ability of diffusion tensor imaging to detect microstructural changes that are happening in the tissue before it's progressed to a point where it's hyperintense on a T2 weighted scan. Now, the issue here is that DTI, while being very sensitive, it also lacks biological specificity. There's a number of other factors other than just damage to white matter that might change FA and MD. So you might have two perfectly healthy, dense neuronal bundles, but with crossing fibers, which can reduce the directionality in your voxel. And also you might have changes in the macromolecular environment of the voxel. So you might have a proliferation of glial cells, or you might have increased interstitial fluid, and all of these can change your signal as well. So the inference of these microstructural changes in DTI is not always clear. And that's where this study came in, where we used myelin water imaging to instead get a little bit more specific in the interpretation of changes in white matter tissue. If you're not familiar, myelin water imaging is a form of T2 weighted imaging that takes advantage of the fact that the T2 decay of water in the brain is different in different cellular environments. So you can detect two different peaks of T2 decay time. You first see this short peak, using this guy, a short peak and then a longer peak. And the short peak has been histologically validated to correspond to the peaks associated with water that's trapped between the layers of myelin that wrap around an axon. Whereas the longer peak is more related to intra and extracellular water. And so because you have this relationship between this short peak signal and myelin water, you can use these metrics to estimate the density of myelin in the voxel. And this is more specific in terms of its biological interpretation than diffusion tensor imaging. And so we have then this idea that maybe these quantitative neuroimaging techniques might be used to gain more specific insights about tissue characteristics. And so the question now is, how do we apply these in white matter hyperintensities? So that's exactly what we did in this first study. We had 47 older adults and 33 individuals who were in the chronic phase of stroke recovery, meaning they were greater than 6 months after their stroke event. This is a complex figure, but essentially what's important to know here is we segmented the tissue of the brain into white matter hyperintensities, normal appearing white matter, and CSF. And for the individuals who experienced a stroke, we dilated the stroke mask by 10 millimeters and removed it from all subsequent data. So we wanted to avoid any contamination of this data from peri-lesional stroke infarcted tissue. Then we took white matter hyperintensity masks and dilated them in 2 millimeter increments in order to get this spatial distribution around the lesion, and then extracted diffusion tensor imaging and myelin water imaging data from these perilesional regions. So following up on those early DTI studies, we ask if white matter metrics are altered in the white matter hyperintensities and surrounding normal appearing white matter, again, in these 2 millimeter increments of distance from the lesion all the way up to 10 millimeters. So first showing some of the DTI data. So we see FA this pattern exactly replicates what's been seen in previous findings with low FA in the white matter hyperintensity and gradually increasing with distance from the white matter hyperintensity. And the data is plotted separately here by group, older adults versus individuals with chronic stroke. MD, same thing. So high MD in the white matter hyperintensity gradually decreasing. And we start to see a bit of a difference in our stroke and older adult groups as well. Then with myelin water imaging, so first looking at the GMT2, which is that long peak signal, this is our increased intra and extracellular fluid. So same thing that you see with mean diffusivity, indicating increased fluid within the white matter hyperintensity and gradually, and it's also altered in the perilesional tissue, but gradually leveling off with increased distance. And then finally, the myelin water fraction. And here's where the story gets a little more interesting. So in the older adult groups, we see no difference really in the white matter hyperintensity and perilesional tissue. And then in the chronic stroke group, we saw a significant group by distance interaction. And post-hoc contrast revealed that it was within the white matter hyperintensity itself that individuals with chronic stroke had lower myelin water fraction, but there were no alterations in the perilesional tissue. So then diving a little bit deeper, we looked at what could be driving these group-related differences within the white matter hyperintensity lesion. And we did this by relating the imaging characteristics in the lesion to whole brain white matter hyperintensity volume, which is an index of the severity of white matter hyperintensity disease. So what we saw here was that there was a significant relationship with MD and whole brain white matter hyperintensity volume along the x-axis there. And with the myelin water imaging metrics, we saw relationships with GMT2 and myelin water fraction in the lesion itself. And what you can see here is that there's not really a clear differentiation between the older adult and chronic stroke group. So instead, it looks like this continuous pathway of relationship that's driven by the overall size of white matter hyperintensity volumes in the brain, which is an index of the severity of white matter hyperintensity disease. So some takeaways here are, first of all, that white matter hyperintensities as a visible manifestation are probably representing a spectrum of underlying pathology. There's some suggestion from our work then that this is starting from early increases in interstitial fluid and gradually progressing to a loss of myelin with increased severity of the disease. And the reason we make this conclusion is because we didn't see any perilesional changes to myelin water fraction. They were all driven by fluid sensitive metrics, which includes diffusion tensor imaging, as well as the GMT2. And this replicates findings from histological studies, but importantly, this gives us a way to extend them into in vivo neuroimaging analyses. So then we wonder if this might be helpful for disease staging in white matter hyperintensities, and if this later stage demyelination might be another way we can index brain reserve in this context. And an open question, I wish I had an answer to this question, but that's for future work, is, would these imaging measures actually be better markers to explain variability that we see in individual functional status and decline? Open question for us all. So then moving on to concepts of cognitive or functional reserve, I want to start talking about interactions between white matter hyperintensities and stroke lesions. So this is a lovely figure from a recent review article, which is, capturing this idea that you might have this subtle damage that's appearing in our normal white matter with interstitial edema. And then this might be causing more far-reaching consequences for the white matter fibers that are affected, including cortical thinning of remote and connected regions. And then in the event of an acute infarct occurring, these processes are probably all interacting to influence the degree and the patterns of damage that are seen in the brain over time. So a nice recent meta-analysis looked at relationships between white matter hyperintensity burden, which was here graded in 3 levels of severity, and functional outcomes and overall outcomes after stroke. And what they saw in this study was essentially a dose-response relationship between the severity of white matter hyperintensities and negative outcomes. And this was for dementia, for any functional impairment, which they categorized very broadly in the context of this study, for recurrent stroke and for stroke-related mortality. So it's this idea that as you have more concurrent white matter hyperintensities, it seems that stroke outcomes are worse. And so the question then is, how are these white matter hyperintensities potentially interacting with processes of neurological recovery after stroke? And can we understand this from a more resilience-based framework? So this is a study that I'm about to submit for publication, hopefully next week. So upcoming work, you can catch it on a preprint server soon, which I did in partnership with collaborators from the Enigma Stroke Recovery Working Group. And so here, we're bringing in this idea of damage to the corticospinal tract as being an important predictor of motor impairment after stroke. There's been a number of studies that index the degree of damage to the corticospinal tract using various metrics, and it's an extremely consistent predictor of motor impairment, which makes sense because you have this major output pathway of the brain, and if you damage it, you're going to impact your ability to activate your muscles, and this is going to relate to motor impairment. So the index that we used specifically in this study was weighted CST lesion load, which is essentially just a degree of the overlap between your stroke lesion, which is pictured here in orange, and your corticospinal tract pictured in blue. We had 223 individuals who were at least 7 days post-stroke and beyond, this data was collated from 7 international sites. And our aim was to test if the relationship this consistent relationship between motor impairment and corticospinal tract damage is moderated at all from concurrent white matter hyperintensities. So we tested this with an overall beta regression model. And what we saw here, we have CST lesion load along this x-axis, motor impairment along the y, and then white matter hyperintensity volumes in the next panel over. So we saw main effects and relationships using beta regression modeling between the degree of damage to your corticospinal tract and the degree of motor impairment. Higher values here indicate more severe motor impairment. And this is the expected relationship that's been heavily established in the literature at this point. However, we also saw over and above corticospinal tract damage, age, days post-stroke, and stroke lesion volume, a relationship between white matter hyperintensities and motor impairment. However, and this was contrary to our hypothesis, there was no significant interaction between corticospinal tract lesion load and white matter hyperintensity volumes. So what I initially expected to see was that this interaction would indicate that at different levels of white matter hyperintensity volume, you maybe have different directions in the effect of the CST lesion load relationship, but we didn't observe that. So then we asked, rather than being an interactive factor, is it possible that white matter hyperintensity severity is an effect modifier of this relationship? An effect modification here, we're thinking about the concept of different subgroups and whether relationships might vary in different subgroups of the sample. And so we stratified the sample into mild and moderate or severe white matter hyperintensity volumes, and this threshold was determined based on lots of other clinical literature that suggests that this might be one potential functional threshold for white matter hyperintensity induced cognitive impairment. So splitting the data into these two groups, first, importantly, what we saw were there were no group differences in the degree of motor impairment, degree of CST lesion load, or in total stroke volume. So essentially, these groups were fairly similar in terms of the degree, their stroke characteristics, and their motor impairment. However, we did see different effects of the relationship between CST damage and motor impairment in these subgroups. So in the individuals with mild white matter hyperintensity severities, there was a significant relationship between CST damage and motor impairment. However, in our moderate severe individuals, this relationship was attenuated and it was no longer significant. And so what this might be telling us is that as you have more concurrent white matter hyperintensities in the brain, then potentially these become relatively more important in explaining your overall outcome in motor recovery. And so these might represent a neurologic subgroup, or at least something to consider when we're building our prognostic models of motor recovery after stroke. And then just one more study to think about this idea of subtypes in stroke recovery. So this was a really nice recent paper also from the Enigma Stroke Working Group that looked at brain resilience in the context of stroke. And what they saw here, so they had actually a very large sample size for a stroke neuroimaging study because this is Enigma data, so it's pooled across many international collaborators. The full sample in the paper was about 1,000 individuals, which for a stroke neuroimaging data set is pretty good. And in this sub-analysis, what they did was propensity score matching. They matched individuals based on their stroke lesion characteristics and their degree of brain pad, which in this study, brain pad is a machine learning derived metric that gives an estimated age of your brain based on structural neuroimaging characteristics. So this will be impacted by white matter hyperintensities, cerebral small vessel disease, cortical atrophy. It's a nice overall measure of essentially how old does your brain look relative to how old should it look based on your biological age. And when they looked at brain pad, what they saw was the color scales here indicate the probability of better outcome, and the shapes also indicate probability of better outcome. And these values that are connected based on lines are indicating the matched pairs from the propensity score weighting. So essentially, if your stroke lesion characteristics and your age and sex, et cetera, are held equivalent, then your brain age, brain pad in this study, is predicting whether you have a good, which is at the bottom here, or poor outcome after stroke. So this is our sort of direct resilience measure. So as you have more concurrent age-related damage, brain atrophy, et cetera, this is a really heterogeneous measure that could be impacted by a lot of different factors. You have either vulnerability to the brain to a stroke lesion or potentially resilience. For the sake of time, I'm actually going to move past this guy. So just some other ideas. All the data that I showed so far is cross-sectional, but obviously longitudinal studies are really essential in order for us to try to identify how pre-existing cerebrovascular disease close to the time of the infarct goes on to impact trajectories of recovery. I'm also thinking a lot about, loops or cycles or ongoing changes that might occur over how the white matter hyperintensities or cerebrovascular disease might impact as well the ongoing processes of neurodegeneration, of Wallerian degeneration, et cetera, that are happening in a stroke-affected brain. And then just kind of introducing an idea, in the concept of disease comorbidities, something that I also wonder about is how these things then interact with behavior. So here we have an example from individuals who might have diabetes. So diabetes being strongly associated with white matter hyperintensities and changes to the central nervous system. But you also have changes to the peripheral nervous system from diabetic neuropathy for similar mechanisms. And so the question that I have, and I think this might be a good group to ask this question is it possible that something like this, where you not only have these central nervous system changes that are going to impact your feed-forward drive to your motor system, but you also have changes to your peripheral nervous system, which is then going to impact the ability to sense and plan and give information back to your brain. How might something like this impact motor behavior, impact your ability to engage in rehab, impact the types of benefits that your brain receives from behavior? I think there's a lot of potential feedback loops and cycles that are ongoing when we think about resilience in this capacity. So lots of different factors that might be impacting variability in clinical presentation and course, not only the size of your lesions, but where are they located? What are other resilience mechanisms that are occurring in the brain? And this idea here of comorbid conditions, things like diabetes, hypertension, et cetera. How are those also impacting brain health or your behavioral capacity in order to maintain good function over time? And I'll just propose that neuroimaging is really nice in this context because it might help us understand all of these different processes and potentially develop markers that could be used in clinical settings in order to measure interactions between brain damage and resilience. So as the last thing, I'm just going to switch a little bit now and talk about some of my postdoctoral work, which I'm doing at the BCCDC. The context is a little different, but it is all related to overall patterns of health and aging. So I'm doing an embedded research project with representatives from various health authorities and officials at levels of government in BC and BC Cancer as well. We're using administrative data records, so the BC Chronic Disease Registry and Cancer Registry, in order to look at patterns of co-occurring diseases across the population. And specifically in this study, we have all residents of BC aged 20 and up who are logged in these administrative databases from 2001 all the way to 2020. The reason that the Ministry of Health and BC CDC are interested in this question is that we know that our population is aging and medical needs are becoming more complex. And as you have more co-occurring conditions, you're increasing dramatically the complexity of healthcare. So you're going to experience more fragmented care. You're going to potentially have more adverse drug events. Individuals with more chronic conditions are more likely to be hospitalized and have increased mortality and loss of functional independence. And of course, we can think of all of this from a brain resilience and health framework as well. Here's a list of the conditions that are present in our registries. So what you can notice is a lot of them are really relevant for the concepts we've been covering so far. So we have stroke logged in the registry. We also have neurological conditions like Parkinson's disease, Alzheimer's disease and other dementias, diabetes, heart failure, hypertension, ischemic heart disease, etc. We also have a number of cancer subtypes. We talk a lot about our aging population, and we also need to start considering our increasing prevalence of chronic disease in the population. So here's data from our multimorbidity analyses. So what we're doing is just looking at how many people in BC have 2 or more of any of the conditions on that list. So in 2001, this is age standardized prevalence. So these numbers are adjusted for changing population demographics. 2001, there was about 17% of the BC population that had 2 or more chronic conditions, and now it's almost a quarter. And this prevalence changes dramatically with age. So as you get older, you're more likely to have 2 or more chronic conditions, and in fact, more than half of older adults in BC are managing more than one chronic condition. And strikingly, for our oldest members of our population, the majority have 4 or more chronic conditions. So a dramatically increased complexity of care in the context of aging. So the primary goal of our project is to mine patterns of co-occurring chronic diseases. And we're doing this with a network-based approach that is very familiar to us in neuroscience, but is less utilized in certain contexts in epidemiology, although it is used a lot in disease modeling in terms of contagious disease spread. So in our disease networks, each disease is conceptualized as a node in the network. We arrange, just to orient you to the plots I'm about to show you, we arrange our diseases based on broad disease categories. And then the size of the nodes that you'll see represents the prevalence of single diseases. So smaller means that single disease has a lower prevalence in population, and larger means it's higher. And these diseases are connected by links. And in the plots I'm going to show you, these links are representing the prevalence of the diseases co-occurring in combination. So thinner and lighter lines represent less prevalent disease combinations, and thicker and darker lines represent more prevalent combinations. So here's some disease networks from our oldest adults in the fiscal year 2019-2020. So these are BC residents aged 80 and up. I have the plots split by sex here, with females on the left and males on the right. And so what we see is a really complex network, a complex pattern of relationships between chronic diseases. So I'll just orient you here a little bit. So we have, for instance, our cardiometabolic diseases all sort of arranged together here: heart failure, hypertension, ischemic heart disease, stroke, and diabetes. And then we have some neurodegenerative diseases to highlight up here. So Alzheimer's and related dementias and Parkinson's. So there's a lot of patterns that are not as expected in these plots. So for instance, more prevalent single diseases also form more prevalent disease pairs. But this also highlights just the idea that we're not just talking about single diseases, we're not even talking about disease comorbidities. We're talking about often multiple disease patterns and relationships that are quite common and highly prevalent in our older populations. So, the highest and thickest lines here are indicating a prevalence of 42 out of 100. So 42% of the population showing some of these highly prevalent disease patterns. So looking at those networks is nice, but ultimately we also want to apply clustering algorithms in order to move from just pairwise disease combinations to try to identify higher order patterns that are occurring. The clustering algorithms that we identify is grouping diseases based on the links, the unique links between diseases. So any disease node can have membership in multiple clusters. And the links are grouped based on their similarity and strength relationship with other diseases. So if you think of a social network analogy, we would be grouped together if we talked more of the same people, essentially. If you have more people in common, you'll get grouped together. And from this, we can extract some clusters, which represent, again, those higher order disease patterns in the population. So showing a first group of clusters here, and sorry, my text animation is wrong at the bottom there. I put in some manual labels because I find the abbreviations are kind of a nightmare to look at after a while. So up top, here's a disease cluster that emerged in females and a similar cluster that emerged in males. We often have anywhere from, I'll say, 4 to 8 clusters that emerge in each of our population subgroups we're looking at, and some are quite similar between sexes and others are not. So here's one example of one that might be similar. So in the female disease cluster, we have a lot of conditions that relate really to sort of fundamental cardiometabolic underpinnings. So you have diabetes, chronic kidney disease being another end-stage complication, common complication of diabetes, gout, which has a lot of inflammatory connections, and then our heart failure, ischemic heart disease, and stroke. And you can see that this network is also highly interconnected between all of these nodes. Then in the males, we have similar diseases emerge, but the relationships between diseases are different, and it's a little bit of a sparser network of relationships in males. And then just showing another example of 2 clusters that appeared that are similar, but still not perfectly equivalent between the sexes. And the reason I'm highlighting this one is because this is where some of our neurological diseases were linked. So we have Parkinson's disease and dementia, and also schizophrenia, mood and anxiety disorders, and stroke. And so again, this is indicating potentially some of these fundamental cardiometabolic health processes that are underpinning probably the emergence of specific diseases and their relationships between one another. And so the reason that I think the clustering approach and this network-based approach is interesting is because this might let you reveal some synergistic interactions. So there's a lot of work in the multimorbidity or comorbidity space, which shows that the burden of individual diseases is not additive. It's often multiplicative or interactive on health outcomes. So having more diseases, it's not just a case of individual impacts added together. The outcome impact on health, the impact on outcomes is typically worse than you would expect. But this also might represent opportunities for positive interventions and positive synergies. So if you can try to interrupt chains of disease progression, then you can, with a single intervention, potentially prevent multiple future ones. And if you know what those patterns are in a predictive manner, then you might be able to be really targeted in some of the interventions you deliver. So next steps for this project, this is hot off the presses, as they say. So analyses are very much ongoing. The next step is to incorporate temporality as a primary outcome here in order to really try to then mine. What I'd like to look at is trajectories into stroke and trajectories into dementia, and also comorbidities that occur after the incident events of those conditions. And in terms of a long-term vision, I see a lot of potential to integrate these sorts of analyses and concepts with our neuroimaging markers of brain health to have sort of an overall measure of cerebrovascular health and brain resilience. So this, this idea of synergistic interventions and preventative medicine, it's not new. It's something that gets a lot of attention. There's a nice infographic from the American Stroke Association. So we know, there's a number of factors that increase risk of any of these cardiovascular metabolic conditions. And then conversely, that means there's behavioral and lifestyle interventions you can take to try to mitigate or prevent the progression of these diseases. And, I also like to think about this in terms of overall frameworks of health and resilience. So not only the individual lifestyle or risk-related factors that might impact the development of or the recovery from some of these negative diseases associated with aging, but also, the social context and the environmental and natural context in which we're all embedded. And, you know, especially here, this idea of healthcare access, who can access care, who can actually be seen by a doctor early enough that you can be flagged for preventative intervention, and who's able to, you know, make their rehab appointments, who's able to have someone looking out for them in the community, et cetera. All of these things might not change some of the neurological factors that we talked about at the beginning of this talk, but they certainly could change your decline over time. And I'll just leave with one thought here. So this was a really recent paper that came out, a review paper published in Hypertension. And this was really interesting to me because I hadn't seen it before. So this is specifically talking about white matter hyperintensities, but there's emerging evidence that all the way to the intrauterine environment, there are factors that are setting up a higher propensity for cardiovascular disease later in life. And so when we're thinking about how we can promote resilient brains and healthy aging, we might have to think really far back, you know, all the way back to our early life and the early impacts of our brain, and how then these can also create our cycles and our loops over time and manifest ultimately in health or disease. So many people to thank, of course, and I'm also looking forward to your questions.
Great, thank you. Got the mic back there. Take questions from both the in-present audience as well as the virtual audience. So, see one now. Just reach a second. Please, questions.
All right.
Yeah.
Oh, if I can open the chat.
All right, thank you. That was brilliant. I really especially like the network maps of like disease clusters. That's just a super clear way of seeing that. It's really exciting. I'm wondering about when you start building in temporality, which is also very exciting. Whether you have some way of looking at kind of hormonal fluctuations, particularly with the female sample set, like kind of the effects of menopause, or if you can infer that from any of the data that you have available.
That's an excellent question. So we're working with administrative data records, and essentially the disease level categories that we draw are based on prescriptions that you had filled. They're based on hospital admission claims. They're based on MSP claims. So that's sort of the level of information that we have about the population. And we also know date of birth, and we know sex. So we could potentially have a typical age of menopause onset with some uncertainty around it. And you could look at transitions around menopause that way. But unfortunately, we don't have direct measures of individual hormonal status. But really interesting question, obviously something that's really important to consider in the context of brain health.
Sounds good. Thank you.
Yeah.
I have a follow-up question actually to that. Just getting back to the same study. You did separation between sex. Did you also look at the demographics, like sort of the demographic information, cultures, ethnic origins, and that sort of as well, because it's quite a rich data set that way. Possibly look at those.
Yeah. So we currently don't have ethics access for things like racial and ethnic categories. We do have high level of geographic specificity. So we have data aggregated all the way down to the level of CHSAs, and we can know some of the demographic characteristics of those different geographies. But yeah, also something important and potentially something we could include in the future, but currently we don't have the access. And really to do that work well, we need to make sure we build partners with people who can help us, who can use their expertise to guide those types of analyses. So I would really like to have those partnerships in place, definitely see that as a future direction, but not currently.
Is this on, by the way?
Yeah.
I just did push it. Oops, give me a second. Yeah, we're good. Sorry about that. Likewise, another question on yet the same study. From a methodological aspect, how were you differentiating between your different clusters?
Yeah, so that's based on the, we're using something called the link community detection algorithm developed by On in 2010. And so the clusters are developed first by, it's a hierarchical clustering analysis based on link similarity. The link similarity is determined by the neighboring nodes for each unique cluster link or each node link. And so the algorithm actually cuts the hierarchical cluster analysis at a level that maximizes the density in the distribution. So it's all essentially done within the model itself. And we're not making any decisions manually about where to cut our dendrogram in order to derive our clusters.
Okay, cool.
Yeah.
Thanks. I wonder if you could elaborate on the functional and cognitive reserve. If I could use the analogy, if I were to use 50% of my muscular capacity to do my daily activities, I can afford to lose 20% of the capacity and still, depending on how lose it, have a potential for rehabilitation and carrying out my daily activity. Is that in that context, that cognitive, and if so, how do you measure it? How do you measure that capacity?
I think maybe we could think of that in terms of brain reserve potentially. Like if you are strong or if you have more potential strength in your muscles, then there might be less effort that's required in order to accomplish the task. And so that an equivalent of that might be, you know, having your early life environment building up cognitive reserve or just avoiding any negative determinants of brain health, so that then if an insult occurs, you have further to fall before you have impaired function.
How do you measure that? How do you measure that reserve?
Yeah.
Quantify that. Sorry.
Yeah, I think, you know, the white matter hyperintensities are potentially one way, or something like the predicted brain age is potentially one way you could measure almost the inverse. You could say that if you have less white matter hyperintensity, you probably have more healthy white matter. That's one potential metric. But, you know, there's many others, and there's work that's even just looking at connectivity profiles and connectivity in the brain and how that might relate to the capacity. But, you know, it's really an active area of research right now. I don't think we have a good answer for what is a good marker of reserve neurologically.
One more question, actually, prior to go back to one of your slides on the spinal trauma. And there was, when you did the stratification by white matter hyperintensity load, was we confused on that scatter plot?
This guy? Yeah.
Yeah. So the moderate to severe show more motor impairment or less motor impairment with a larger CST?
Yeah, I was thinking around this as well. So I think the way that we can make sense of this is really the uncertainty of the estimate, that there's potentially more variability being captured from white matter hyperintensities in the model in our severe group, that's then reducing the explanatory variability of the CST lesion load. But I know what you're getting at, in that it's almost the opposite of what I would have expected to see, which is that at a higher level of lesion load, you would have less motor impairment in the mild group. And so since we...
Sorry, go ahead.
Oh, no, please, yeah.
I was going to say, I wonder if because we don't have a good index for how long the white matter lesions have been there. So it could be that by having this, kind of gets back to the brain reserve thing, is it could be by having multiple small lesions that could be asymptomatic, you start building more resilience. So that if you have, this is, you have to test it right, but if someone has sort of mild, doesn't have anything at all, so they get a stroke, oh my God, everything shuts down. Whereas someone that has multiple infarcts or multiple white matter lesions, they may have built more resilience.
It's,
to deal with that.
Yeah, it's possible. That's sort of the idea of ischemic preconditioning. It's something that is seen, you know, even at the level of like, you know, doing cuffs to your arm and seeing that there's a brief period of hypoxia can sometimes improve resilience in tissues. I think it's possible, but equally it might be possible that having these areas with disrupted brain vasculature and disrupted brain flow, could that then lead to accelerated white matter hyperintensity over the course of stroke recovery? So I could see it going both directions, and ultimately we're going to need longitudinal samples to ask that question.
Was there any effect of the proximity of the white matter lesions to the CST?
Didn't test that, but it's a nice idea. Yeah.
So I know a lot of the, well, at least in MS, the white matter correlation symptoms is not very clear a lot of times.
Mhm.
I don't know if that's true in this in general. The spatial distribution of white matter hyperintensities tends to not really correlate with the particular symptoms you might be showing.
Yeah, there's some work, from Marco during his group and others, which are looking at sort of the idea of strategic lesion locations with white matter hyperintensities. And the thing that is interesting, I think, with white matter hyperintensities for this question, just gonna move back to another slide here. You have a pretty, not perfectly predictable, but kind of a characteristic pattern and characteristic regions, white matter tracts that might be more or less effective. And especially here, looking at this, you think about transcallosal tracts. And so is it possible then that some of the cognitive profile that you see in white matter hyperintensities is not only representing the size of lesion, but the location and the fact that they tend to happen in the same white matter tracts over time? And certainly with the stroke literature, something like CST lesion load is an independent predictor of motor impairment above whole brain stroke volume. So if we can find something similar, a strategic tract, then we could maybe do a much better job with our modeling. And yeah, I think it's a really important question. I did a study looking at anterior thalamic radiations and trail making tests, so processing speed in older adults and individuals with chronic stroke. And we found that white matter hyperintensities in anterior thalamic radiations related to trail making abilities. So, and it might also depend on the cognitive domain you're testing too. Yeah.
We have a question online.
Yeah, so we have a question from Zoom from Brian Greeley. The question is related to demographics question. Do you have behavioral data or ADLs for those comorbidity clusters? Specifically, do you know the level of functional reserve for those people? And I wonder how those clusters may change for people with high functional reserve for those who don't.
Yeah, again, another thing that I wish we had with our administrative data sets. So we really do have medications, hospitalizations, and MSP. But I think with the development of so many nice longitudinal survey-based data sets and large data banks and brain banks that are being developed over time, there's a lot of potential to apply this sort of network-based multimorbidity context with those other really important measures of health status in order to answer those types of questions. So limited currently, but I think there's a lot of opportunity in that area.